So if we look at the current workflow that we have today, a urologist performs a biopsy, we send it to the pathologist, they fix it in formalin and paraffin embedded, and then over a period of a couple of days, in 4 to 7, it might be quicker at certain places, but on an average, 4 to 7 days, you get a pathology report that generates the GLEASON score, or the GLEASON, the grade, if you will. We then can incorporate genomics into this decision-making process on patients with prostate cancer. We take those paraffin-embedded specimens, we send them off to a commercial lab, wherever the lab is, whether it's Salt Lake City or in San Francisco, in Redwood City, and essentially can do a genomic profile, look at a number of genes with the oncotype, it's the profile that I talked about earlier this morning, and then we can identify a certain genomic pattern to try to predict aggressiveness or adverse pathology. Well, what I'm suggesting we might want to do is something totally different. We might want to take a biopsy, and what we've developed is a technique that we can actually grow out live prostate cancer cells. So we take a biopsy, we can put it in a test tube, send it to a laboratory with a proprietary extracellular matrix, grow out single cells, then teach machine vision algorithms, develop machine vision algorithms and learning, and then create different matrices to then predict, risk stratification. And I'll go into this in a little more detail. So I use this picture, and this is from Ashok Chandler's daughter, to illustrate the difference between genomic markers and phenotypic markers. So the snapshot, if I take my iPhone out and take a picture of his daughter, you can see she seems to be very happy. Good picture, she's happy, she likes the environment. I send the picture to you and you say, there's a happy daughter. And that's what a genomic marker does. It gives us an idea of what the tumor biology is at that certain particular time. But what a phenotypic marker is here you can see is the picture, she was happy, but all of a sudden her father says, no, you can't do this, and you see a change in the environment that she's in. She's not such a happy girl now. Maybe daddy took the candy away or won't let her play, but she's not a happy person. And that's the difference between genomic markers and phenotypic markers. And I'm going to show how we apply this concept, if you will, into science. So we know that there's tumor heterogeneity that exists, and indeed that provides and really causes a challenge for us. There's intra-patient variation, there's intra-patient variation, and intra-tumor variation that exists when we talk about prostate cancer cells. But we can take phenotypic markers, both cellular and molecular. On the cellular side, we look at morphology, dynamics of adhesion and spreading and cell stiffness. On the molecular side, we can look at protein expression, subcellular protein localization, and so on. And we can look at a panel of markers or a platform, much like Larry's alluded to earlier, of 600 markers to really try to understand how this tumor is behaving and reacting in the environment that exists. So this is how this test is done. We take a biopsy, we get live tissue, essentially, and I'll show you the study that we did after robotic prostatectomies, place it in a test tube, send it off to the laboratory in Boston. We put it in a cell loading and put it in what we call a microfluidic device. It's essentially a plastic tray in which we put a proprietary extracellular matrix. Then we're able, you'll see in a moment, to grow out single cell monolayers, and then using live and fixed cell imaging, we can quantify these biomarkers using machine learning and machine vision algorithms, do statistical algorithms, and then develop what we call predictive matrices. And an example of these predictive matrices are this general adverse pathology potential, local adverse, and metastatic adverse pathology potential. And I'll talk about those in detail, but they're essentially the things that pathologists look for. Is there seminal vesicle invasion, extraprostatic extension, and so on and so forth. So here is the first question that you want to answer is, can we grow out prostate cancer cells? And we've been able to do this. And here you can see is looking at a series of controls compared to the extracellular matrix, and you can see that there's much more robust cell growth in this extracellular matrix across the board. Now, when you look at this over time and you see, are we able to really sustain growth? And indeed, both with normal prostate and cancer tissue, we're able to do that. And you can see that these curves across the board show really good growth across the board. So here's now what we've done is we've labeled these prostate cancer cells, and we're going to study 600 biomarkers on each one of these. So each number represents a single cell, and we're looking at that cancer cell and how it behaves in its environment. And we can then use automated machine vision software to generate statistical algorithms, and then we can go and develop predictive matrices from that. So we looked at both live and fixed cell phenotypic markers. You can see the live and the fixed cells, and then with machine vision and machine learning, we're able to develop risk stratification from this. So as I said, here you can see the samples are taken from live prostate cancer. We send it to the laboratory, and we did this with robotic prostatectomy patients. So we knew where the tumors were, and in the study I'm going to show you, we biopsied not only the prostate cancer, and that was confirmed by the pathologist, but we biopsied normal tissue in the prostate because we were curious to see if there's any kind of a field effect. When we think about tests like MDX confirm, it's really based on this field effect that there's something abnormal around the tumor that may be and provide information for us. We're able to grow the cells out, and then we can use this automated imaging, and we may see in the future, we may not need the pathologist as much as we rely on them today. So here are some of the matrices that we've developed. Essentially, they're, the general adverse pathology potential combines with local and metastatic adverse pathology potential, and these are clinically relevant factors that we're using today to try to understand prostate cancer. So here's a study that we did with a couple of other institutions. We essentially took radical prostatectomy specimens, biopsied them, sent live prostate cancer tissue, put it in this extracellular matrix, put it in a microfluidic device, and then looked at the phenotypic biomarkers. Over 600 biomarkers were studied in each prostate cancer cell that we grew out. And here you can see we had mostly Gleason 6 and 7s, 3+4s and 4+3s. We did have some higher grade cancers, but the majority of this was really to try to differentiate, can we separate the Gleason 6s and the Gleason 7s? Because the 7s are starting to be the significant tumors that we really want to treat. And here's what I alluded to earlier. When we look at how predictive these are, you can see that the Gleason score is about 60 to 70%, both in the sensitivity and specificity. Genomics is about 70 to 80%. But the phenotypic markers, when you look at the areas under the curve, are much, much higher in comparison to these other studies. Here you can see is the breakdown of these 237 specimens, the sensitivities across the board when we look at these matrices and sensitivity, and the areas under the curve all seem to be better than what we have at the present time today. So here is a real practical application of this technology. You can see here are two Gleason scores that are illustrated. The blue score represents a Gleason 3+3 tumor that has no adverse pathology. And you can see it scores low on a MAPP and a LAPP score. Looking at the orange dot, this is one Gleason 3+3 tumor that has one adverse risk pathology feature associated with it. And indeed, you can see the MAPP and the LAPP scores are much higher across the board. So indeed, when we look at the sensitivity, we can also use this to predict upgrading and biochemical recurrence. And the areas under the curve, as I've said, are really quite high and really can give us a lot more information. Now, when we did this study, we thought about this tumor effect and this field effect. Is there something that can be gotten from a tumor, from normal tissue that may predict prostate cancer? So what we did is we biopsied normal tissue, which was confirmed by the pathologist under the microscope, and then we ran it through this, essentially growing it out in the extracellular matrix, and ran it through our phenotypic analysis. And what we found is that we were able to see, and these patients all had tumor, that there was a very high sensitivity and specificity area under the curve. Now, interestingly enough, there weren't a lot of specimens, only 87 specimens, but it's food for thought that may provide some interesting information. The other component of this technology or platform is this machine learning. We have biomarker data, we have machine learning, and then we're able to develop a threshold and then look at predictive matrices. These are the biomarkers that are objectively ranked. When you start to see, compared to these others, we do very, very well. And you can see that when you combine both tumor and field effect, it's about 0.9 to 0.98 areas under the curve. So we're able to grow out prostate cancer cells. We can quantify it. We get some very interesting data. And our next study is going to be a biopsy study where we take a fresh biopsy and run this technology through it. Thank you.